Chapter 5, Kinetoplasta, Trypanosomes and their kin, Part 6, Trypanosoma cruzii. Now here with Trypanosoma cruzii, we're looking at a disease referred to as Chagas disease. It's named after the doctor who initially recorded it, characterized it, and the first one to isolate what they thought was the potential causative organism. This is a New World disease, okay, Central and South America. It is related to Old World Trypanosoma brucei and the sleeping sickness within the same genus. Here in the New World, Central South America, we see there have been quite a few people infected. This has been a very understated public health issue. In the 90s, in Central South America, they think anywhere from 12 to 19 million people. That's between 1 and 2 million people a year were infected. Nowadays, it's about 561,000, so about 500,000. When you look here, you see that this is the life cycle of trypanosomes. The Kidvidid, I can never say that, Kidvid bug, the kissing bug, which is a insect parasite of not only us humans, but many other mammals, sloth, possum, armadillos, and whatnot in Central and South America. Basically warm-blooded mammals. The kissing bug goes in for a blood meal. Now, unlike the Trypanosoma brucei, the Old World, with the tsetse fly and it injected with saliva the parasite into the new mammalian host. Here what we find is the Redivivid bug, when it goes in for a meal, as blood goes in, feces comes out, and in the feces will be passed out trypamastogotes. Trypamastogotes, which will then travel the very short distance from the feces to the open wound, which was the feeding site for the kissing bug. Once in, then you start to see it travel through the bloodstream, entering into different tissues and whatnot. The trypamastogotes will be found circulating in the mammalian bloodstream, in our bloodstream. Okay, posterior end is going to be pointed. The anterior end is going to be a little more rounded off, so a slightly different morphology than what's been seen and discussed with brucei. The flagellum, instead of being folded back and attached to the plasma membrane, it's actually going to be a free flagellum. So it's out there. It does still have a narrow undulating membrane, so it does still have that sail-like membrane. It's just that it's not attached to a flagellum. And it's also going to have its own kinetoplast that is slightly located to a different place than with what was seen in brucei. The amastogotes for cruzii are going to develop in muscles and other tissues, highly vascularized tissues. Trypamastogotes exit out, the capillary beds become the amastogotes, which take on a more spherical, spheroid shape, and they're going to occur in clusters. So as they go through asexual reproduction, the new daughter cells, the new resulting cells, are going to stay clustered together. So there is quite a bit of differences between cruzii and brucei, not just the morphology, but when we look at the biology, we look at the metabolism. Turns out cruzii is not quite as metabolically active. What I mean by that is it can't use quite a few of the carbon sources, the sugar sources that brucei does. When you see that it's going to go through and start doing anaerobic fermentation, which is something that the trypanosome genus can do, cruzi is not able to complete all the steps that brucei was. I'm sorry, yeah, the brucei was. So there's going to be a lack of total energy ATP production that you see brucei is able to do, and thus some of its metabolic byproducts, its metabolic waste products, are going to be different. This is a clinical diagnostic issue that we have been able to exploit and can take trypanosomes of unknown species and not only can we, as we're going to see in a few slides, do DNA testing and whatnot, but we can also do metabolic testing. Can it, yes, no, ferment certain sugars? Can it complete the consuming of a carbon source and release of CO2. When it comes to the pathogenesis, part of what makes cruzi such an issue is it starts off with a localized inflammatory response due to the metacyclic forms. Okay, remember when the metacyclic for all trypanosomes is what's actually going to be infective. So it's the metacyclic trypamastogote, which is that phase that can get into human blood or the human tissues in the human blood and be infective. What we're going to see is within a week or two from the localized inflammatory site, there's going to be a slow spread to the regional lymph nodes. Once there, Trypanosoma cruzi is going to want to be taken up by the innate immune cells, primarily macrophages. Okay, macrophages are going to phagocytize, try to take up and kill Trypanosoma cruzi, but they will be unable to complete the phagolysosome fusion event, which would lead to digestion of anything not you that was taken up by the phagocytic cell. Inside the macrophage, you'll start to see rounds and rounds of asexual reproduction. Inside the cell that's supposed to be taking it up and killing it. So it's a perfect place to be, protected from other innate immune cells and protected from the adaptive immune response. So it's an intracellular parasite for part of its life cycle. What's going to happen here is that it's going to start to develop what is called a pseudocyst. You're going to have the cell enlarge, 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 burst, neighboring cells get infected, intracellular pathogen, intracellular parasite, and other cells are going to come and start glomming on, and it's going to start to wall off. It's going to start to keep everything there in one place. When you look here at this picture, you'll notice that weird elongated shape there that's full of lots and lots of little nuclei. That's the pseudocyst in a cardiac muscle cell. Okay, it's not just going to be in the macrophages. Any cell that this can get in, or any area can get in and wall off, going to get this pseudocyst. Since it's a cyst, it's going to be, you're going to be able to feel a small nodule, depending upon the size and complexity. It may even show up on some x-rays. The pseudocyst will continue to increase in size as the asexual reproduction continues until it hits a certain point where it's just going to burst, and you're going to get dozens, if not hundreds, of trypamastogotes released all at once. They're going to be flooding through the bloodstream. This is where you're going to see the parasitemia. so that this is now the point where the parasite in the blood is going to be at its highest concentrations. It's going to spread within minutes of the pseudocyst bursting, and you're going to start to find the cruzi invading tissues all over the body. When it comes to the Chagas disease, you're going to see that there are two phases. Okay, first one's going to be an acute phase. Okay, this is right after the inoculation of the parasite. Okay, the kissing bug comes in for its meal, passes out the feces. The metacyclic trypomastigote enters in through that small microscopic open wound. Going to get the inflammatory response. For many patients, going to see the development of a shigoma. Shigoma is going to be a small red nodule at the site of, the wound. The inflammatory response is causing that to get, bigger and enlarged. For some people, due to the site where the site is, got to remember the revidavid bug is called the kissing bug because it has it seems to preferentially like to go for the blood meals in the face, primarily around the mouth and sinuses, the nose, because it's highly vascularized there and the skin is at its thinnest and most pliable. But if it gets up too close to the eye, what would be a shigoma anywhere else on the body, it's going to be referred to as Romana's sign. The eye is actually going to swell up, going to get edema, swelling of the eyelid and the conjunctiva, which is the mucosal lining of the eye socket. So all the tissues here are going to get red, swollen, and discolored. This will, over time, start to decrease, go away. As the trypomastigote starts to spread, pseudocyst, burst, parasitemia, you then enter into the chronic phase. Okay. Once you have the parasitemia, the cruzi is now spreading through the bloodstream. It's going everywhere. It's also going to the capillaries that are nourishing, that are bringing in nutrients and whatnot to the meninges. You're going to get the trypomastigotes able to penetrate through the meninges, and you get into the central nervous system. Okay, it's not always, once you hit the central nervous system, you're going to have different, just like we saw with sleeping sickness, but it's not quite as progressive as quickly. It's a little slower. The acute phase is a more longer duration with cruzi than it is with brucii. What we do see with cruzi that you don't see with brucii is that there are other nerves that can become involved besides just the central nervous system. The autonomic ganglia also seems to be a targeted tissue. Turns out trypanosomes does have some tropism. Remember, tropism is a pathogen, bacteria, virus, parasite, seems to be drawn to one type of cell, one type of tissue, one type of organ over others. Well, turns out that the autonomic ganglia, remember that's that's how your brain talks to your stomach, talks to your throat and whatnot, so that your brain, unbeknownst to you, can control the muscle contractions for peristalsis starting at your throat all the way down towards your anus. Well, because the autonomic nervous system is being interrupted, that impulse doesn't go through. So the muscles of the throat aren't contracting that rhythmic contraction as needed for you to be able to swallow. Peristalsis of the stomach, of the intestines, doesn't occur. The muscles start to atrophy. It gets harder and harder to swallow food. Normally, the tube, that's your esophagus, should be like this. But because of the atrophy, it starts to thin, and the lumen gets a greater diameter as the muscles atrophy and the muscles start to thin. The muscular walls of the esophagus, as well as the gastrointestinal tract, start to thin out as the muscles start to atrophy and start to decrease in mass. You end up with what is referred to as megaesophagus and megacolon. As you look here at this picture on your left, you'll see up at the top right, that's what your esophagus should look like. Very highly vascularized, highly muscularized, so that as you go to swallow, stuff gets pushed down through. Same with the small intestine, the large intestine. But as you can see, start up at the left and you go clockwise, you can see that the walls, the muscle tissue starts to thin out, atrophy, and it starts to swell and get much, much bigger. You would think that as the tube gets bigger and things should be able to slide through much easier, and they don't. Turns out that the food and whatnot just can't pass through. It gets caught up. You need the muscles there to help push it along. When it comes to the immunological response to cruzi, there is a bit of an issue. It's an intracellular parasite. It wants to hide inside of our cells. Hard to mount an immune response to a parasite that's hiding in our cells. When it's extracellular, flowing in the blood, flowing in the tissues, yes, macrophages, neutrophils can bind to it, grab it, phagocytize it. Yes, if there's enough during the parasitemia, the spread, when they're extracellular and the waste products are getting released, yes, given enough time, B cells can learn, and B cells can be activated, become plasma cells, start making antibodies to cruzi proteins. The problem is you can't mount a very successful immunological response, and it's not because of, like we saw with brucei, the variable surface antigens. No, here it's because it's hiding in our cells. Antibodies don't penetrate into our cells, so antibodies can't bind to the parasites. Other immune cells cannot kill it if it's hiding in one of said cells. So the only time the immune response can be helpful is when cruzi bursts out of the pseudocyst or bursts out of macrophage and it's extracellular for a few minutes, for a few hours. But that's rarely long enough for, a good immune response to occur. Diagnosis of cruzi, well, blood. Do a blood smear on a slide. Cerebral spinal fluid. Do a lumbar puncture and put some of the fluid, cerebral spinal fluid, on a slide and look for the parasite, the trypomastogote-shaped parasites. You can do it with lymphatics, or you can just take a tissue biopsy. Look for pseudocysts. There are some reports in South America that they have a rather interesting way of doing this. They will take blood or cerebral spinal fluid or whatnot, and they will literally feed it to laboratory-raised insects. So they will put it out as a food source. These blood-eating insects will come in for their blood meal, or they'll just let the patient, here, go in for a blood meal. And they will see. They will wait and see if the insects, they will sacrifice the insects because the insects are a great incubator for the different trypomastogote stages. They will take known uninfected insects, allow them to feed either on a blood sample or on the patient itself under controlled conditions, and then they will wait and see if cruzi develops in the gut, the gastro, the alimentary tract of that insect. They're now working on more immunodiagnostic assays, looking for mechanisms to test for antibodies to Trypanosoma cruzi. Limited, it's been limited there. because the immunoassays, from what they found, it's antibodies against trypanosoma, but they're having difficulty getting successful tests that say, here are antibodies to trypanosoma cruzi, species specific, and not here's antibodies to trypanosoma genus. That's been the issue so far. What they found also is when it comes to treatment, there's the issue that it's an intracellular pathogen. Any drugs you take now, right this moment, today as a patient, will only affect extracellular parasites that are flowing in the blood. And that's only a small portion of the population of the trypanosoma cruzi flowing through the patient's, in the patient right now, because the majority of them are going to be intracellular, whether in macrophages, muscle cells, wherever.